So there are some spontaneous events, basically natural chemical events that happen in the cell in terms of chemical interactions that can cause mutations to happen. And one of them is called tautomeric shifts. The nitrogenous bases, thymine, guanine, cytosine, and adenine, have the ability to switch into their tautomeric forms, which are called the rare forms. And so most of the time they're found in their common forms, shown here on the left, but briefly and occasionally they'll switch into their rare forms, shown here on the right. And what this will usually lead to, if this shift happens right at the moment of replication, meaning right when that enzyme is looking, for example, at the T and deciding what base to put into the new strand, if the T is on the template strand, then if it shifts into its rare form right at that moment, then the enzyme might put the wrong base into the new strand. And that's one cause of mutation. But these naturally occurring events are considered spontaneous, or they create what are called spontaneous mutations. There's nothing you can do to prevent them. It's not caused by some kind of external environmental thing. This is an example. If cytosine switches to its rare form, then, for example, the enzyme will look at that and it'll put an adenine across from it in the new strand. So you'd end up with an adenine where you should have had a guanine. Okay, so that would be also an example of a transition. Thymine in its common form, guanine in its rare form will cause thymine to be inserted across from it. And so should have been a C, but now it's a T, so that's also a transition. But it just causes the enzyme, the DNA replication enzyme, polymerase, to add the wrong nucleotide if the base in the template strand shifts into its rare form right at that moment. And there's nothing that you can do to prevent this. So this is just showing you how ultimately you'd end up with, if one of the strands gets a shift, then you can get a chromosome that has a mutation here. If the wild type chromosome has a TA, then the mutant strand has a CG in that same position. You can also have replication errors, like the expansion of the trinucleotides, for example. This is an even more simple one. You could have like a long stretch of T's in the template, and then during replication you could get this kind of loop out thing that could happen, and you get an extra that's give you an insertion. Or if the template strand loops out, then you could get a deletion in the new strand. So that can also give you insertions and deletions just from replication errors like this. Unequal crossing over, we looked at in a previous chapter. That's a spontaneous event that causes deletions and insertions, really of any length. But there are things that are chemicals that are called mutagens, and these are external things that come in from the environment, and they can cause an increased rate of mutation as well. One example is fibromouracil, which mispairs. If it gets into your cell, it mispairs. It's an analog of thymine, and it will tend to mispair with guanine, so it'll cause an insertion of the wrong nucleotide in the strand. And when you have something a mutation that's caused by a chemical or something that's an external cause, then those are called induced mutations. Induced mutations have a specific environmental cause. They're not spontaneous, meaning they're not naturally occurring mistakes in the cell. Here's some other types of mutagens. EMS is a mutagen. Nitrous acid is a mutagen. Hydroxylamine is a mutagen. These all cause induced mutations, mostly by causing mispairing in the DNA during replication. One thing that's interesting is mustard gas, like EMS, mustard gas is also an alkylating agent. It causes DNA mutation, and it was used in World War I for chemical warfare reasons, because it produces chemical burns and blisters. So they would spray it, and if you didn't have a gas mask, it would burn the inside of your lining of your throat and lungs and so on, and it was really damaging. It also is a mutagen, though, and so what they found is that, 40, 50 years later, those same individuals who were in the battle not only got these burns, but they also ended up with, certain types of cancer because it mutated the DNA in their cells. Acridine dyes are mutagens. They intercalate into the DNA double helix. Intercalate means insert. And they bend the backbone of the DNA. You can see this DNA is kind of warped, and it tends to lead to mutations or mistakes during DNA replication. Ethidium bromide is a dye that's used in electrophoresis gels, and it's very carcinogenic because if it gets into your skin cells, for example, it will intercalate into the DNA and lead to mutations. The only mutation that you're really concerned with is a mutation in a cancer gene, but it can lead to that. One thing we saw in lab is that UV creates pyrimidine dimers. Pyrimidine dimers are covalent connections between adjacent pyrimidine bases. Adjacent means on the same strand. It can't be across on the other strand because you wouldn't have two thymines in a base pair. But next to each other on the same strand, it creates a covalent bond directly between the pyrimidine rings. And that causes the backbone to bend, and if it's not repaired, it leads to mistakes during replication. So, when we're thinking about mutagens, chemicals or radiation that causes mutation, what comes to mind is a test called the Ames Test. And this is used really to determine, among other things, how mutagenic a new drug is. Because you wouldn't want to develop a new pharmaceutical drug, spend a lot of money and time researching it, if you found out in the end that it was a mutagen, because mutagens are cancer-causing compounds. And so the Ames test is a method to determine how mutagenic any substance is, and it's a screen, which means it's a way of cheaply and easily and quickly getting some results on how mutagenic a chemical is. The way that you do the Ames test is you start with bacteria that are His minus. That means they can't make their own histidine. And histidine is an amino acid, so they need that. And so His minus bacteria are plated on a minimal media, agar plates, that don't have histidine. So therefore, those bacteria should not grow. And so I give you this note. Only His plus cells can grow in minimal media, so the His minus cells should not grow. But you're going to add compound X, that's the drug you're interested in testing, to the agar plate. It can be added directly to the agar, or it's on a disc. You put that on the plate. And what you're looking for are colonies that appear on the plate. Because if you get colonies that are His plus, that means that you've had mutation. And those colonies can be either reversions, or actually they could also be suppressions. But any colony that is now able to grow has undergone some kind of mutation to lead to that ability to grow. They do add liver enzymes also to the plates because in hindsight they realized that some drugs that do not appear to be mutagenic are converted into mutagenic substances when the liver enzymes process them in the body. And so in hindsight, they realized that they needed to add liver enzymes to this test in order to check for that effect. But in any case, if you have high levels of reversion back to His plus cells, in other words, if you get a lot of colonies on your plate, then that means that your compound X is a mutagen. So the way they do this is they take the His minus bacteria. On one plate, they just plate the His minus bacteria on plain old plates that lack histidine. That's the control. On the right side, they're plating the bacteria in the presence of the chemical that they're testing plus liver enzymes on the plates that lack histidine. And what you'll get on the control plate, you might get 1 colony, for example. And that's just a spontaneous mutation that you might get. And so that's kind of your background mutation rate. On the plate where you got where you added the drug that you're testing, you might get 1 colony, or you might get, in this example, you have 10 colonies. If you got 10 colonies on this plate where you've added your drug chemical, what it means is that it's causing a significant amount of mutations, and so it's probably a carcinogen. So this result is what you're not wanting to get, because this means that your drug is going to cause cancer. But most of the time when you test a new drug, you'd probably just get 1, colony, something similar to the control, and that means that your drug is not creating any additional mutation rate above and beyond the background mutation rate that would naturally happen. So this slide shows an example of kind of the rare result, but the result that you would get if your drug is a mutagen.